Hi, I'm here. Yeah, I'll just go ahead and share my screen.
Yes, please.
Yeah. Okay. Should be visible now, I hope. Yeah, great. Okay.
You can see it.
Okay. So hi, I'm Bhaskar, and I'll be talking about this topic that will be the architecture of the genotype-phenotype map and the coevolution of complexity. This work was done as a part of my master's thesis at the Indian Institute of Science, along with Dr. Zaman, who is at the Ecology and Evolutionary Biology Department in University of Michigan. So the title of the presentation mentions 4 different slightly terms that need some explanation. The first one is architecture, which sort of indicates some sort of structure in the system. The genotype, which is the set of all the genomic information present inside the organism. And there's the phenotype. So phenotype usually is seen as a sort of traits, like a phenotype is usually seen as a trait of the organism. But here when we say phenotype, we mean a combination of all the traits present in the organism. So a phenotype will be, for example, comprised of whether an organism is tall or short, plus whether it's, for example, it has purple flowers or white flowers and so on, rather than individual characteristics. That's a phenotype. And the first thing I'll go into in the presentation in detail in the presentation is complexity. So what is complexity?
Your slides are not. Yeah, sorry.
Yeah. So this is a broad definition of complexity I'm giving. That is, the phenotypic complexity of an organism is the information obtained about the genotype of the organism when you observe its phenotype. So what do I mean by the information obtained? So on the left side, I have a full genotype space that is available to an organism to explore for a population to explore through mutations and evolution. On the right, I have marked some phenotypes in the genotype space. For example, the phenotype 1 has a certain fraction of the genotype space under it, and the number of genotypes in this space is G1. So when you observe a phenotype, it restricts the possible genotype space of an organism. And you can say that the information obtained about the organism on observing a phenotype is equal to the decrease in entropy of the possible state space. So this definition is pretty well worked out in this paper by Andreas Wegner, and we're using the same definition to define phenotypic complexity. So for example, for phenotype 1, the phenotypic complexity is log G minus log G1. So it's the same as the decrease in the size of the state space when you observe the phenotype. This definition does require the presence of a proper, like a complete model of the biology in the system. That is, given a genotype, you should be able to tell what phenotype it belongs to. So we are also assuming that. So now this definition of phenotypic complexity has some particular interesting features. The first one is that is correlated with the rarity of traits. For example, if the phenotype 1 has a part of genotype space G1, and this space is bigger than G2, then in that case, the complexity of phenotype G1 is smaller than the complexity of phenotype G2. That is, the more rare a trait is in the genotype space, the more complex it is. The second interesting part is that it's usually correlated with algorithmic complexity. So if you might be aware of AVIDA, which is like a digital evolution framework. And on the left side, I've put 2 tables. The first column in the table is the AVIDA definition of a task called NOT, which is algorithmically very less complex. So it has only one NAND instruction in the genome. Whereas on the right, you have an AND task, which requires multiple NAND instructions in the genome, in this case 2. So if we specify both these tasks, the NOT task requires 3 less instructions in the genome. In that respect, you can have 1 of 32 other instructions in these 3 extra places. So the number of genotype sequences that can encode for NOT are at least 32 into 32 into 32 times the number of genomic sequences that can encode for AND. So in that way, the complexity of NOT is less than the complexity of AND. Or the phenotypic complexity is now correlated with the algorithmic complexity of the task, which is another property of the definition of phenotypic complexity we're using here. So overall, phenotypic complexity is the measure of rarity of phenotypes in the genotype space. Now, there's an interesting observation that comes up. So this is from a 2014 paper by Lewis. That is, when you introduce parasites into a evolving host population, it leads to a spontaneous increase in the complexity of the host. For example, if you look at the bottom curve, the host in isolation, it leads to some basal level of complexity in the genome. And this complexity is the algorithmic complexity. So this is the number of NAND instructions in the genome that are leading to a particular task. However, if you were to add parasites to the system, the complexity increases. So now you get more and more complex genomic tasks in the genome. And as soon as you have to remove the parasites in the middle of the run, the complexity decreases again, which is interesting. So why does this happen? What is the mechanism behind this? So the parasites follow a phenotype matching model. That is, in this case, the parasites are trying to match the phenotype of the host in order to infect them. And if you look at this figure, you can see that initially when there are no parasites, there is some fitness complexity correlation on the genotype space. However, when you add the parasites, it occupies a certain part of the complexity space, and it does not allow the host to survive that well because it decreases the fitness. And that way, when you have multiple parasites, the entire curve shifts rightwards. That is, now the hosts have to move and occupy more and more complex tasks in order to survive. So what is the intuition hypothesis behind this work? The first one is each genotype in the genotype space has a corresponding phenotype, and less accessible phenotypes are likely to be safe harbors for the host population, where they can escape the effect of the parasites and survive. So the major hypothesis we have is the positioning of these phenotypes in the genotype space must determine the coevolutionary outcomes, or the structure of the genotype-phenotype map must determine the evolutionary endpoint in these populations. So now the big question is, what do we mean by the structure of a genotype-phenotype map, and what do we mean by coevolutionary outcomes? So structure can be, there can be multiple measures to measure the structure of a particular genotype-phenotype map. The two we are using is skew and neighborhood heterogeneity. When I say skew, skew is a statistical property of the genotype-phenotype map, whereas neighborhood heterogeneity actually takes into account the network structure. And I'll go into these in more detail. So first, I'll talk about the structure itself. So on the left side, you have a three-dimensional hypercube consisting of bit strings of length 3. So each of these bit strings can be thought of as a genotype. And these genotypes can then have a certain phenotypes. For example, these 4 genotypes have a phenotype 0. These 3 genotypes have phenotype 1, and so on. And then I arrange these genotypes into clusters of phenotypes and order them in decreasing order of number of genotypes. So if you remember the definition of complexity from earlier, this, as you go from left to right in this map, the complexity of the phenotype increases. Yeah. So the first property that we measure for the genotype-phenotype maps is the skew. So skew is a statistical property. It basically calculates how skewed the number distribution of genotypes is under these different phenotypes. For example, the genotype-phenotype map on the left side is it's not very skewed because almost all the phenotypes have a similar number of genotypes. Where the genotype-phenotype map on the right side is extremely skewed because the lowest complexity phenotype has a lot of genotypes under it, whereas the highest complexity phenotype is only one genotype under it. And we measure this as a Shannon entropy of the distribution of genotypes under different phenotypes, so that's straightforward. It is not a quantification of the network structure itself because it does not take into account the edges. I should mention that the edges are single mutation edges. For example, on the left side here, these edges connect single mutant neighbors in the genotype space. The second measure we look at is the neighborhood heterogeneity, which takes into account in some respect, the network structure of the genotype-phenotype map. So the neighborhood heterogeneity calculates how diverse are the phenotypes accessible from a single genotype in the genotype space. So to calculate this, we basically look at every node in the genotype space. We look at the phenotypes it connects to. For example, this node will connect to phenotype 2, and it connects to 3 nodes. 3 of these edges connect to phenotype 0, and so on. And then we calculate the Shannon entropy of this distribution of edges for each node, and then we average it over all the nodes. So the minimum value will be reached when all single mutants for all genotypes have the same phenotype. That is, for each genotype node, if you were to make a single mutation, it leads to the same phenotype. And the maximum value is reached when it's equally distributed between all these phenotypes. And you can also think of neighborhood heterogeneity as a sort of evolvability measure, because more genotype-phenotype maps with more neighborhood heterogeneity will lead to more different phenotypes on mutations. So if you make a single mutation, you'll get more. You have more chances of getting a different phenotype, and so on. So these are the 2 measures we are using in this work. What about the coevolutionary outcomes? So the first coevolutionary outcome is obvious, that is the mean complexity, which is what the title states. We are also looking at the phenotypic heterogeneity, which is how diverse the population is when you add parasites to it. So phenotype of complexity I've already described. It is just the log of the number of genotypes in the genotype space minus log of the genotype space under a particular phenotype. And to calculate the average population complexity, we just average it over all the organisms in the population. The phenotypic diversity or heterogeneity is just the Shannon entropy of the population distribution of the organisms in the population. And to simulate populations over these genotype-phenotype maps, we use a very simple Gillespie exact SSA with different reactions. So there's a reaction in which a host is giving birth to another host. Host plus parasite give two parasites, but the host dies in the process. The parasites are also decaying at a rate c, which is as in they cannot survive on their own in the environment. And then there are these transition terms. So for example, a host I can turn to a host j with a particular probability in each step. And to calculate these probabilities, we use the genotype transition matrix. So for each genotype in the genotype space, you can have a transition rate depending on the mutation rate, per site mutation rate. And from this genotype transition matrix and a partitioning scheme matrix, which is just the which describes the genotype-phenotype map itself. So for example, phenotype 0 has genotype 0 and 1 under it. Phenotype 1 has genotype 2 under it, and so on. And we take a convolution of these and we form what is called the phenotype transition matrix, which basically sums up the individual terms. So for example, if phenotype 0 had G0, G2, and G5, there's a genotype under it, we'll sum over the probability, and then we take a column, we normalize it column-wise. So now each term in the phenotype transition matrix contains the probability of transition from one phenotype to other in a single step of the simulation. And we use some particular parameter values, as shown here, which we chose by performing these performing runs with multiple different parameter values and choosing the one which gave steady population at the end of the run. So now, when you think about it, these genotype-phenotype maps have a very trivial state called a neutral state. So if we were not to add a parasite into the mix, and assuming that all phenotypes are the same fitness, so it's a neutral model, the genotype space is relatively small. That is, over the time scale of the simulation, the organisms can scale every genotype in the model, and the population carrying capacity is much larger than the size of the genotype space. And we are also assuming there are no parasites present. In that case, the population relaxes to a state called a neutral state, such that the distribution of organisms among phenotypes mirrors the distribution of genotypes between these phenotypes on the genotype-phenotype map. That is, the fraction of organisms under a phenotype I is equal to the fraction of the genotype space occupied by that phenotype. And in this case, all these three measures become equal. That is, the population heterogeneity is exactly equal to the average complexity of the population in the absence of parasites, which is called C0, is equal to the negative of the skew of the GP map. So now what do we see when we do this? So to perform the experiment, generate random genotype-phenotype maps using uniform sampling of the genotype-phenotype map space. We simulate co-evolving host and parasite population on these genotype-phenotype maps. So we are assuming that both the host and the parasite have the same genotype-phenotype map. And then we calculate population measures on the resulting population at the end of the run. So the first result we find is parasites lead to an escalation in host population complexity, which was what we expected because that's already been seen in previous work. So if you don't add any parasites to the system, the host goes to a particular basal level of complexity, which you call C0. But if you were to add a parasite and you let both these populations evolve together, the complexity increases, and it's somewhere above the basal complexity. The second result we find is the basal complexity on a genotype-phenotype map, that is complexity evolved without the presence of parasites in the system, is equal to the magnitude of the skew. And this again is expected because in the absence of parasite, the system is expected to be in the neutral state. That is, the distribution of organisms among the phenotypes is equivalent to distribution of genotypes among different phenotypes. And that's when you see this. So this is the y is equal to minus x line that denotes that negative of the skew is equal to the mean host complexity without parasites. And all these genotype-phenotype maps give population that lie along this line. The third result we find is, which is the most interesting result, I think, the skew of the genotype-phenotype map positively affects the evolved phenotypic complexity. That is, so on the left side, I have a figure where we have calculated the complexity evolved in the presence of parasite divided by the complexity evolved in the absence of parasites. And as you increase the skew of the genotype-phenotype map, you see that this escalation in complexity is higher. So the more skewed the genotype-phenotype map is, more likely are you to get an extremely high increase in complexity on addition of parasites. And I've also plotted just the complexity before taking a ratio with the basal complexity on the right, along with the skew of the genotype-phenotype map. We don't find such clear results with neighborhood heterogeneity. We see that when you increase the neighborhood heterogeneity of the genotype-phenotype map, the outcomes in terms of complexity are more variable. But then this could also be the result of the fact that the space is sampled more on the right side. So for example, you can see here that there are more points on the with high neighborhood heterogeneity. So this is not a conclusive result. The fifth and another interesting result we find is the host population diversity at a steady state is dependent on the structure of the GP map. That is, so this is the skew line. This is the line you would expect the host population diversity to lie along if there were no parasites in the system. However, when you add a parasite, the entire distribution shifts upwards, and the deviance from this skew line is increased as the skew of the genotype-phenotype map is increased. So again, the more skewed the genotype-phenotype map is, more even is the distribution of organism among different phenotypes than is expected from the neutral state of the genotype-phenotype map. So what are the final results that we find? The first one is the genotype-phenotype map with higher skews promote an increased escalation in phenotypic complexity. That is, if you have a more skewed genotype-phenotype map and you add parasites to the system, the average rarity of the traits increases. The average rarity of the traits increases for more skewed maps. Host population generally evolve more phenotypic diversity than expected in the presence of parasites. So in the presence of parasites, they evolve a higher level of diversity compared to the neutral state. And this is slightly inconclusive in this case, but genotype-phenotype maps with high neighborhood heterogeneity or evolvability have a more variable outcome in terms of mean complexity. So there are certain limitations and assumptions we're making here. The first one is that these results are likely not to hold for very large genotype spaces, because you are assuming that in the simulation time, the mutational process can explore every point in the genotype space, which is not true for large genotype spaces. However, if you want to look at, for example, the bacteriophage receptors in bacteria, this might be a valid model for cases like that, which is a subset of the entire genotype. The second part is the coarse graining to phenotype transition takes away some of the structure of the network, because you are going from a genotype transition matrix to a phenotype transition matrix. You might be taking away some part of the network structure. And we have recently stumbled upon a way to prevent this from happening. So we have found a way to simulate each individual genotype now, and we will be working on that soon. So what is the final conclusion of the entire presentation? So the escalation in complexity in a host population or introduction of parasites can be an evolutionary standard. That is, while it might be dependent on selective features, selective factors, it's also likely dependent on the structure of the genotype-phenotype map itself. It's a property that arises out of the structural constraints on the system rather than direct selection for increased complexity of hosts. Yeah, and with that, I'll conclude my presentation. Thank you.
Thank you very much, Bhaskar. Very nice talk. Please, if you have questions, put them in the question and answer section. And either I read out your question, or if you raise your hand in the participants, then you can we can probably unmute you and give you the video. Just to warm up, maybe I ask a first question. So I really enjoyed how the clarity of your methodology and the clarity of the results you're getting. And of course, there are many variations of this basic model that you can perform, a basic simulation. For example, do you have you explored, or would you have an hypothesis what might happen if you go to more complex population structures, in the sense that you have multiple parasites, or so essentially more co-evolving species in your simulation?
Yeah, so right now we're looking at only 10 different phenotypes, but that number, so the problem there was because we were modeling individual transitions between genotypes, it takes a lot of time to calculate all the transition terms in the matrix. But we recently come up with a way to avoid doing that. And I think once we have that, we'll be able to do even more, increase the number of species.
But in terms of having more than only a host and a parasite, but maybe different predator-prey populations with more than the predator and the prey. So, for example, multiple predators.
Yeah. So if you, I think you're talking about having not having just two types of species in the bipartite interaction network, for example, but having say 3 or 4 or different types. That can be added to the simulation. I think it's not. It should be straightforward considering how simple it is, but we have not tried that yet.
All right. And there's a question by Yan Kim. Can this approach be generalized to a system in which there is a phenotypic plasticity? So if one genome can generate various phenotypes depending on environmental conditions?
Yeah. So firstly, there are no environmental conditions in the system, so that can be modeled itself. But I think the major problem there would be there is no organism life cycle as such. each organism exists like there's no life cycle, so you cannot have some sort of developmental paths working on top of this system. However, maybe using Avida, you can model something like this much easier, because Avida actually talks about you go into the individual life cycles as well. So in that case, it'll be easier to model phenotypic plasticity, I think.
But maybe with not mapping each genotype on a single phenotype, you could just have a, to some extent, stochastic distribution of this particular genotype can be 50% probability matched to one phenotype and 50% to another. That would be a way to incorporate it in your model without going into the complexity of Avida.
Yeah, I think that's a great idea. Yeah.
And another question by Jeff Nitschke. What is the example of a skewed genotype-phenotype map in biological systems?
Yeah, so I have been thinking about this in terms of bacterial receptor on bacterial surfaces that bind to bacteriophages. So, for example, if you have a certain docking region where the bacteriophage receptor docks, changing certain parts of the protein coding structure might not change the docking, the interaction energy, and it might not lead to infection. Whereas if you have if you're changing another peptide, for example, amino acid, for example, it might lead to a more drastic change in the binding affinity, for example. In that respect, you can have certain parts of the genome which are more susceptible to, which lead to more susceptible change in phenotype, whereas some others might not. So yeah, I think in general, all genotype-phenotype maps that you think of will be skewed to some extent. We have not really looked at like real genotype-phenotype maps, because that will also be an interesting thing to look at. So how skewed are real genotype-phenotype maps, for example?
Great, thank you again, Bhaskar. And if there are more questions, please continue to just use the question and answer mode, and Bhaskar can take your questions typed. All right, and then we proceed to the last talk of the section, which is by Dragana Laketic on the hyperdescriptions of quantum dynamics, a case study in avian magnetoreception. Dragana, will this be a live presentation, or is there a video being played?
Yes, I would suggest that we play the video recorded, because my Ubuntu has become a bit unstable. I had to log in through Xorg, Windows, and a Wayland to make Zoom work correctly. So I think it's safer version to play the video.
I can relate to you, because I also got my Zoom crashed in the first part of the session. So
yeah, it happens to me now.
The video.
Okay.